Hello and good afternoon friends, welcome to CEC EduSet live lecture. Dear friends, in yesterday's lecture we discussed on history of immunology, and today we would be specifically talking about immunity and vaccines. And for this we have again with us in our studios Dr. Hardeep Kaur. She is an assistant professor in Department of Zoology, Ramjas College, University of Delhi. So first of all I would like to welcome our guest Dr. Hardeep Kaur. Welcome to the EduSet lecture, ma'am. Yesterday we discussed a lot about immunology. Basically, we studied about how this term coined and how the various facts came into existence. But today, vaccines. When we talk about immunity, of course, vaccines appears to our mind, and there appears a question: what are the vaccines available?
Yeah,
so over to you, ma'am.
Yeah, thank you so much, Kitika. It's a hot thing to know more about the vaccines and what is the current study going on in immunology. So, coming first to what immunology was and is, we had done a lot about it in my yesterday's lecture. I will just say that immune system is just like any other system of our body, be it a reproductive system or a respiratory system. It has series of cells, it has tissues, it has organs. These cells tend to communicate with each other. So, in a way it is very analogous to the nervous system also, wherein there is a communication between cells and organs. Here again you have cytokines which actually became a messenger between cells of the immune system. So, immune system typically is just like any other body system. As I told you yesterday, we only come to know of a system if it goes wrong, if it goes away. So, if something goes wrong, you have a viral attack, you have a bacterial infection, then only if you feel, oh, something is wrong with my immune system. Till that time, we are happily sitting merrily with all our systems and we hardly know what is happening in our body. So, many of the pathogens are invading our body, be it virus, be it fungi, be it bacteria. They are invading our body from any place, and all these cells of the immune system, they form a first line of defense like phagocytes. They will just engulf the bacteria or virus which is coming inside our body. Now, the question arises is, what if the system becomes hyperactive or hypoactive? Both thing is deleterious to our existence. If it is hypoactive, that means it is not acting in a nice way, there is an immunodeficiency, that is also which is not right. So, if it is hyperactive, for example, a pathogen which is a very probably very normal pathogen like a pollen grain, which does not become pathogenic in most of the individuals, become pathogenic for a particular person, and a person becomes hypersensitive to that pathogen. That also is again not a very nice situation to take place. So, immune system has a great responsibility. It has to strike a perfect balance. It cannot go hypo, it cannot go hyper. It has to be at the right place. So, with that background, I would like to come to my real topic, that is immunity and vaccines. Now, immunity, as we described yesterday also, is a state of protection from infectious diseases. It can be non-specific, wherein it can be the immunity or the immune system is functioning against any pathogen. It could be viral, bacterial, it does not matter which pathogen is invading our body, it is acting on that. So, immunity could be non-specific or it could have specific components. So, based on what kind of immunity we are talking about, we further divide this immunity as innate immunity and adaptive immunity. Now, as you can see in this slide, here we are showing immunity is of two types, the innate immunity and the adaptive immunity. We can further characterize adaptive immunity as active and passive, and the active can be further characterized as natural active or artificial active, or natural passive or natural active, artificial active. Now, here the nomenclature we are following is adaptive being categorized into natural and artificial, and natural further can be of active type or passive type. Similarly, artificial can again be of two types, active or passive. Now, what is an innate immunity? Innate immunity is the immunity with which a person is born with. You have not done anything to improve or spoil or improve the type of immunity you are born with. It is It is specified by your genes. It is a natural resistance with which a person is born. It is non-specific in nature, that is, it protects against any kind of pathogen irrespective of its species or type. It could be a virus, it could be a parasite, it could be a fungi, it could be a bacteria. It is actually the first line of defense. It does not demarcate whether it has to give a better immunity against a particular pathogen or a less better immunity, I mean less than the what is the best for the other one. So technically it is not demarcating between any pathogen, it is non-specific in nature. Whatever pathogen is coming, it finds it's pathogenic, it just gives a response to it. So it's a first line of defense, and most of the cells like phagocytes, mast cells, dendritic cells all come in this category where they give a response, they phagocytose the pathogen and hence remove the pathogen. Now, it is mainly comprising of 4 defensive barriers: the anatomical barrier, physiological barrier, phagocytic barrier and inflammatory barrier. Now, first coming to the anatomical barrier. The anatomical barrier that forms a barrier to the pathogen is the skin and the mucous membranes. The skin is the most important anatomical barrier. It is composed of sweat as well as oil glands. You have certain natural flora both inside as well as on the skin surface which prevents the entry of the pathogen. If there is a cut, that means your skin is not intact, that becomes an area through which a pathogen can invade a body. So, anatomical barrier, an intact skin along with its mucous membrane, along with its sweat glands, along with its oil gland, forms a perfect barrier to the pathogen, the first barrier to the pathogen and the first type of innate immunity. The physiological barrier, that means a change in body temperature and pH. Now, supposedly, if the pathogen has invaded the body, the leukocytes present in the body will detect these invading microbes and they will signal the brain to increase the body temperature to create a non-ambient environment. See, most of the pathogens have an ambient temperature of development at 37 degrees Celsius, which is a normal body temperature. Now, if you increase the body temperature, the pathogen might itself die. As we see, whenever the temperature of our environment increases, most of the viruses die on their own. So, similarly, analogous situation takes place in our body wherein you increase the body temperature and the pathogen which is invading the body dies on its own. That becomes a physiological barrier where leukocytes play a very important role. Then comes the phagocytic barrier. We have phagocytes like macrophages, we have natural killer cells. Their main duty is to devour or phagocytose the pathogen the moment a pathogen is realized to have entered or invaded our body. These phagocytes just reach the point of entry of the pathogen, engulf the pathogen and kill it by the enzymes present inside. So, natural killer cells or phagocytes or dendritic cells for that matter, they are the main important cells of the innate immune system. And of course, there is an inflammatory barrier. Whenever, for example, there is not an intact skin, there is a break in the skin, there is a cut in the skin or sometimes the skin gets burnt, that becomes a point of entry for a microbe. Now, you will find whenever there is a cut, there is a redness, there is a heat, there is a pain, there is a swelling at that region. All these signs are showing that the healing process has actually begun at that point. So, all these barriers, whether it is anatomical, whether it is physiological, whether it is phagocytic or the inflammatory barrier, they are the 4 important barriers that play a very important role in the first line of defense in innate immunity. And as already said, innate immunity, you cannot do much about it. It is your genes that define your innate immunity. If you are born with good genes, you have a better innate immunity. If otherwise, you can't do much about it. Now, types of innate immunity. There are 3 basic types of innate immunity: the species innate immunity, the racial innate immunity and the individual innate immunity. When we define the species immunity, it is the immunity that is possessed by all members of a species. For example, if I say I am a Homo sapien, I am a human being. So, belonging to a species of Homo sapiens, I am protected from all plant pathogens. So, naturally all these plant pathogens, for example, a tobacco mosaic virus, no tobacco mosaic virus infects human. So, technically all those plant pathogen that the plants get infected with, they will not infect us or Homo sapiens because we belong to a different species. So, in a way we have got an immunity towards a certain pathogen because we belong to a particular species. That is called species specific innate immunity. Now, coming to the racial immunity. Now, within the same species you have different races that will show difference in their susceptibility to a pathogen. For example, within the species of sheep, the Algerian sheep are supposed to be resistant to anthrax. Within human beings, if you consider, we have people suffering from sickle cell anemia. These people do not get afflicted with malarial parasite. The reason being, the reasons are different, but this is a typical example of racial immunity where people suffering from sickle cell anemia are immune to the malarial parasite or malarial pathogen. That is called a racial immunity, that you belong to a particular race within the species and hence are protected from certain pathogens. That becomes a racial innate immunity. And of course, the individual innate immunity, wherein which is very similar, where I had just described, that different individuals belonging to the same species could have variable immunity. Here of course the genes are responsible. A person is different innate immunity wise from the B person. So, they have difference in immunity because they bear different genes which are responsible for their immune system. So, immune system here is defined by, or the innate immune system with which this individual is born is defined by the genes he or she is carrying. Now, this slide particularly shows the innate immunity. The first is the epithelial cells which are forming the barrier. Then of course comes the cells like phagocytes, dendritic cells, plasma protein, natural killer cells or NK cells. If you see the timeline, between 0 to 12 hours is the time, let us say hypothetically 0 is the time when the pathogen has invaded. So, the innate immune system comes into function. Now, for the first few hours it is the innate immunity that is functioning. Now, most of the times if innate immunity is not sufficient or probably these cells are not capable of removing this pathogen from our body, then the adaptive immunity or adaptive immune system comes into play. So, technically it is a defeat of innate immunity that probably, although one can say that adaptive immunity is actually going along with innate immunity, but innate immunity is the one with which a person starts with to protect itself from a pathogen, and this role is taken over by the adaptive immune system. And you can see from day 1 to day 5 onwards, it is the adaptive immunity which is actually playing a major role in protecting an organism from the pathogen. For example, if you will see in this slide, there is a naive B cell which, because of a pathogen, this the red one is the pathogen here, because of the pathogen gets primed by the pathogen and start producing a specific antibody against that pathogen. More and more antibodies are hence produced which finally kills this pathogen. Similarly, in this slide if you see, there is a naive T cell which on coming in contact with a cell carrying a pathogen becomes primed, shows its receptors, the T cell receptors, and becomes an effective T cell which finally kills the pathogen. If it is a T cytotoxic cell, it will become cytotoxic T lymphocyte and directly kill the pathogen, or if it is a T helper cell, it will provide help to the B cell to produce specific antibodies and hence kill the pathogen. So, technically adaptive immunity is a stronger immunity than the innate immunity. It is something where our immune system is working. In innate immunity, our immune system is not directly working. However, in the adaptive immunity, our immune system is actively working to remove the pathogen. Now, we can define adaptive immunity as it is an important part of the immune system. It is a protection from an infectious disease agent that is mediated by B and T lymphocytes following exposure to specific antigen, and characterized by immunological memory. That is very, very important. Not only our immunity in this case of adaptive immunity is specific to the pathogen. For example, as I told you, in case of innate immunity, there is no specificity. The innate immunity is acting against any pathogen, be it a virus, fungi, A, B, C, D, Z. But in adaptive immunity, the immunity that is produced is extremely specific and it has got immunological memory. This immunity is not present at birth. It is also called specific immunity, as I told you, because it tailors its attack to a specific antigen which is previously encountered or may not be encountered, and it has an ability to learn, adapt and remember. So, this ability is very, very important for adaptive immunity. This is what makes it very important, because once it has worked against a particular pathogen, it memorizes that pathogen, and when that pathogen comes back again, it mounts a much bigger response. That is what is adaptive immunity. Now, these are few of the properties of adaptive immunity. As you can see, it is highly specific. That means it ensures that distinct antigens give specific responses. That means antigen is recognized, remembered. It is highly specific to a particular antigen, unlike the innate immunity which had no specificity. Diversity. It enables the immune system to respond to a large number of antigens. It could be any type of antigen, and our adaptive immunity has so much of diversity, so the property of diversity that it enables our immune system to respond to so many different types of antigen. There is a memory associated with the adaptive immunity. That means it remembers or memorizes the antigen that has come first. It has recognized that antigen once it has infected a body, invaded a body, and that same antigen, if it enters again, it will mount a much stronger response than the first time. This is called the memory or adaptability, or the memory associated with adaptive immune system, that it recognizes the antigen. Yes, this is the same antigen which has invaded our body first time. So, now I have to combat it in a much stronger manner. So, this property of memory is very, very important property for the adaptive immune system. Then of course comes clonal expansion. Clonal expansion is the fourth property, where once a particular B cell has been motivated or has been primed and it has started clonal expansion. I told you about clonal selection theory in my last lecture, in which the pathogen comes and binds to a particular antibody on the B lymphocyte. This antibody is highly specific to this pathogen or an antigen, and this triggers that particular B lymphocyte to have a clonal expansion. That means more and more cells of that particular B lymphocyte will be produced, more clones of that B lymphocyte is produced, and that B lymphocyte is responsible for producing a particular antibody which is extremely specific to that antigen. So, there is a clonal expansion of a particular B lymphocyte. This ensures that antibody of a particular type is produced. That particular type would be extremely specific to this antigen. So, wastage is removed. Only those antibodies are produced which are very specific to that antigen. This is called clonal selection or clonal expansion. Specialization. It generates responses that are optimal for defense against different types of microbes. That means the response is highly specialized. If particular antigen is coming in your body, then our antibody which is produced is highly specific to that particular antigen. Contraction and homeostasis, that is, it allows immune system to respond to newly encountered antigen. And finally, non-reactivity to self. Now, what if, consider a situation where we are producing an antibody in humongous amount against a particular antigen, but that particular antibody starts acting on your own body cell. That is not a nice situation. Your body cells would be killed in that scene. So, the antibody response or the response that is produced in the adaptive immunity should not be self-reactive. That is a very important part, that it should prevent injury to the host during responses to foreign antigen. There should not be any reactivity to the self antigens or to the self cells. So, all these properties of adaptive immunity or adaptive immune system is very important. And again, as I had been telling you, these properties and this adaptive immunity is one where our immune system is working. We are not born with, I am again reiterating, we are not born with this immunity. Here our immune system is actively functioning and has all these properties. This is called adaptive immunity. As you can see in this slide, in primary and secondary immune responses, there is a specificity and memory in adaptive immunity. Our immune system, if you see, if there is an antigen X, you will see in the primary response there would be an antibody against this antigen X. This is called the primary immune response. Now, if the same antigen X comes again, in the blue part you will see the immune system has mounted such a tremendous antibody response many times than the primary response. Now, if you see here, the antigen Y along with antigen X has come. So, antigen Y is having a primary response. So, at any given time, if 2-3 antigens are coming, if one antigen is coming as a secondary infection, it will have a secondary response. But if another antigen is coming first time into our body, it will have a primary response. This is the way how the adaptive immune system plays its role in our body. Now, adaptive system includes both humoral immunity components as well as cell mediated immunity components. What does this humoral immunity means? Humoral immunity, humor actually is the name for the body fluids. Now, we must understand that antibodies which are produced by B lymphocytes. So, let us understand the cells of the adaptive immunity first. So, the most important cells of adaptive immunity are B lymphocytes and T lymphocytes. Now, B lymphocytes produces antibody. These antibodies are technically the receptors present on the B lymphocytes. They can be given off into the bloodstream also, or they can be present on the surface of the B lymphocyte also. Now, when this immunity is present in the body fluids used to be called as humor in the olden days in the Latin. So, this is how the word humoral immunity came into being. Now, as you can see in this slide, there is a microbe which is entering and it is priming a B lymphocyte, or it is triggering a B lymphocyte to produce a antibody against this pathogen. Similarly, if you see the cell mediated immunity, there are two types of T lymphocytes. One is called the T helper cells, the other is called the T cytotoxic cells. The phagocytosed microbe has come inside the body. The microbe has been phagocytosed by a macrophage and it is shown to the helper T lymphocyte. There is a process, there is a MHC requirement, MHC2 requirement if it is a T helper lymphocyte. The T helper lymphocyte in turn will produce a cytokine which will give help to the B lymphocyte to produce a specific antibody against this microbe. Similarly, in case of T cytotoxic lymphocyte, mostly it is our antigen presenting cell. The antigen presenting cell is any cell of your body. It could be a cell which has gone wrong because of tumor development or that has become cancerous, or it could be a cell which is inhabiting a microbe like a virally infected cell, but it is your body cell. This body cell or the antigen presenting cell is bringing the antigen to the T cytotoxic cell. The moment this antigen is brought to T cytotoxic cell, of course, the antigen recognition takes place along with MHC1, and once this pathogen is recognized by the T cytotoxic cell, the T cytotoxic cell becomes develops or transform itself into cytotoxic T lymphocyte or the CTL cells. These CTL cells will further kill this antigen presenting cell along with the antigen. So, antigen gets killed by the direct action of cytotoxic T lymphocyte. So, technically whether it is the B lymphocyte or it is the T lymphocyte, these produce both the humoral immunity as well as the cell mediated immunity, which are the key or the salient features of adaptive immune system. Now, what are the various types of adaptive immunity? Now, types of adaptive or acquired immunity, we also call it as acquired immunity because we acquire it during our lifetime. We are not born with it, as I had been repeatedly telling you. This acquired immunity or adaptive immunity is of two types. One is the naturally acquired immunity, one is the artificially acquired immunity. In some of the books you will see acquired immunity being categorized as active and passive, and then active further categorized as natural or artificial, and similarly passive being categorized as natural and artificial. Here we have categorized acquired immunity as naturally acquired active immunity or naturally acquired passive immunity, similarly artificially acquired active immunity and the passively acquired and the artificially acquired passive immunity. Now coming first to the naturally acquired active immunity, you must understand that first of all let us understand what is the difference between an active and a passive immunity. Now in an active immunity, it is your immune system which is functioning, you know, it is an active functioning of a person's immune system. Here the pathogen has come inside your body, the immune system is now giving a response. Now generally we must understand that there is some titer of antibody always present in our blood. This could be a specific antibody to a pathogen. So naturally whenever a pathogen has entered our body, that particular antibody will combine with the pathogen in a lock and key manner. It is a very tight function between them. Now once it is a specific function rather than a tight one. Now the specific antibody is binding to a pathogen. Now there would not be a free antibody in the blood. So naturally there is a down titer, that means the titer of antibody, free antibody present in the blood gets lowered. So to begin with there would be a latent period, and after that latent period there will be a negative phase in case of active immunity when the measurable immunity is much lower than what it was before the antigenic challenge, because most of the antibodies which are present, which are specific to this antigen, has now combined with an antigen. So the free antigen, the antibody titer has decreased. Now following this beginning after the latent period where the immune system is setting into function, there would be a primary response, as I just showed you in my previous slide. There was a primary response. Now after the primary response has happened, yes, here you can see the antigen has come, the antibody which is present in the titer or blood titer is combining with that specific antigen, and then it is mounting a primary antibody response. Now following that, if the same antigen comes again the secondary time or the second time or a tertiary time or the third time, there would be much bigger response because few of the B lymphocytes have memorized that antigens. Those cells are called memory cells. So technically B lymphocytes are of two types, the memory cells which retain the memory of the antigen, and there are plasma cells which are antibody producing factories. So in a secondary or a tertiary response we have much bigger antibody response as compared to the primary response. Now coming back to our description of active immunity and passive immunity, the active immunity is much longer lasting and there is an abundant secondary response as I just told you. There is again immunological memory associated with active immunity. It remembers or memorizes the antigen which has come. Now it is much more effective and a better protection because it is highly specific. Now passive immunity, though specific, it is specific, but here we are getting a ready-made immunity. Now why do we need a ready-made immunity? See, sometimes the person's immune system is not functioning well. He is immunocompromised or there could be any reason, and he is combating a life-threatening disease. At that time it is more important to save this person. So what do? You provide him with a ready-made immunity. That is what is called passive immunity. There is no latent period because naturally the antibodies which has been provided to this individual immediately acts to kill that pathogen. Naturally there is no negative phase also. But the immunity is transient. It will last only for few days, maybe a week or two. But after that you will have to give another dosage of ready-made immunity. So you will have to provide new and new, but of course you have to save that person from that massive disease at that moment. But with repeated providing this immunity to this person repeatedly, the efficacy decreases and the person become more and more tolerant to this particular antibody, and there is a level of immune elimination. It is, for example, if the first time it has been, the person has been provided with ready-made immunity, after some time, for example, that lasted for 2 weeks or perhaps 3 weeks, 2.5 weeks, after that if you provide the same immunity to this person second time, probably the immune elimination is taking place much earlier, rather than lasting for 2.5 weeks. Now it lasts for probably less than 2 weeks. So this rate of immune elimination increases with repeated dosage or repeated giving of the same antibody to this person. There is no secondary response, there is no immunological memory, and again the good thing about it is it provides an instant immunity, but this immunity diminishes in its effect with time on repetition. Now, as you can see between active and passive immunity, as here it is written, the active immunity is, for example, a microbial antigen. This microbial antigen you might have contracted from another person naturally, or it has been given as a vaccine. It comes inside you, it takes days or weeks, and then you will provide, the body will gives you a protection from this disease, whether this disease is a naturally acquired disease or through a vaccine, and there is a recovery. Of course, the most important thing is there is a specificity, it knows the pathogen, it is the immune response that has been mounted is extremely specific to this pathogen, and it is associated with a memory. There is a memory associated with it, it will remember the pathogen. Now, the second type of immunity, which is the passive immunity, as you can see, we have serum or antibodies from the immunized animal. For example, an animal has been exposed to the same pathogen, that pathogen will lead to immune system working in that. For example, in horse, if you expose a horse to a particular pathogen, the horse will produce the immune system of the horse will produce an antibody against that pathogen. So, the pathogen has got a specific antibody to it. Now, if we take the sera from the horse, take out the antibodies, use them in human, so we have a ready-made source of antibody against that particular pathogen. That is called a passive immunity. Here, as you can see it in the slide, the serum is containing antibodies from an immunized animal. This individual adoptive transfer to the naive animal, and the antibody sera or hyperimmune sera is collected from the animal, given to the individual who require it. This passive immunity, this person gets it, he recovers from that disease. Now, whether this has specificity? Yes, of course it has a specificity, because we know the pathogen, the antibody which has been produced in the animal is specific to that pathogen, but does it have a memory? No. Here we are providing a ready-made immunity to the organism, to the human being. So, naturally it does not have any memory. That is the difference between active and a passive immunization. Now, again coming back to it, the active and passive immunity are again either natural or artificial. For example, when we are talking about natural active immunity, it is the type of immunity where we suffer a disease. For example, by just getting infected by a virus. These days, change of season, you have so many viruses coming, cold, influenza, all these viruses changing their type, and so you get infected with various types of viruses. So, this is a natural occurrence of disease in your body, which leads to triggering of your immune system. A specific immune response is produced, that is called active immunity. Now, in a natural active immunity, your immune system is working hard. Now, there is another kind of natural passive immunity, which, for example, a fetus developing inside the mother during a gestation period gets a ready-made immunity in the form of IgG antibodies through the placenta. Similarly, a child that is born, an infant, for the first 3 months of his life gets ready-made IgA antibodies through the milk that is produced, or through the colostrum, and then followed by the milk that the mother feeds, breastfeeds. So, that is a kind of a ready-made immunity which a child is getting. The child's immune system is not capable to combat the pathogen for the first 3 months of his birth. So during that time, this passive immunity, the natural passive immunity, is extremely important for the baby or for the infant to protect from various different pathogens. So it is a very important type of, but it's a naturally passive immunity which a mother is providing to the baby. Now similarly, there is artificial active immunity where we are imitating the infection. We are either killing the pathogen or making it so weakened, but we are not totally removing the pathogenic capability. I mean, we are not making it pathogen. The pathogenicity is removed, but the pathogen is given to the body in such a way that the body recognizes it as a pathogen and produces a immune response. Either the pathogen is weakened or it is in a less titer, in whatever form we are giving. If it is live, in a less titer; if it is dead, it is attenuated or weakened. Then that pathogen as a vaccine is given to the body mounts an immune response, and when the same pathogen actually infects us, we provide a secondary response and hence are protected. This is the concept behind the vaccination, but this is artificial active immunity. Similarly, there is artificial passive immunity. As I just told you, in artificial passive immunity, sometimes a horse or another animal is exposed to a pathogen and it produces an antibody against that pathogen. The hyperimmune sera from the horse is collected and that is used in the human. Now, it has been found during this artificial passive immunity that sometimes there is a reaction given to this hyperimmune sera from the animals. So, these days we are actually using the hyperimmune sera from other individuals also. Like for example, a person convalescing after a disease, his immune or his sera is collected and this is then used to against another individual who has been inflicted with the same pathogen. So, these are the various types of acquired immunity. Now, the most important, as I was just telling you, is the artificial acquired active immunity, which is through the vaccines, which is through the dead or attenuated pathogens. Now, my further discussion about this is regarding vaccines. Now, vaccines can be called the biological preparations that provide active though artificial acquired immunity to a particular disease. A vaccine typically contains an agent that resembles a disease causing microorganism and is often made from weakened or killed form of the microbe, its toxins or one of its surface proteins. So, we utilize the fact that our body produces a response against a pathogen. So, we utilize that pathogen either by weakening it, by attenuating it, or by taking some part of the body of the pathogen which is immunogenic, or by using some of the compound or the product produced by that pathogen against which our body produces a response, and use it as a vaccine to produce a response in the body and prepare our body in case the same pathogen infects us again, so that we can produce a secondary response at that time. That is what the concept is behind vaccination. Now, as we discussed it yesterday also, the history of vaccination, the term vaccine and vaccination are derived from variolae vaccinae, the term which was devised by Edward Jenner to denote cowpox. We discussed it yesterday also that in the middle of 18th century, while serving his apprenticeship as a surgeon, Edward Jenner learnt of the story that was common in the rural areas, that dairy workers would never have the fatal or disfiguring disease smallpox, because if they had contracted cowpox, then they would not contract smallpox. Now, this was the thing which prompted Edward Jenner to do his further work. We discussed it thoroughly yesterday. In 1796, Jenner took pus from the hand of a milkmaid who was having cowpox and scratched it into the arm of an 8-year-old boy, Phipps. His name was John Phipps. 6 weeks later, he inoculated the boy with smallpox and this boy did not get smallpox. This prompted him to coin this term vaccine. Vaxa means cow. Jenner extended his findings and in 1798 reported that his vaccine was safe in children and in adults. The second generation of vaccines was introduced in 1880s, that is late 19th century, by Louis Pasteur. So, technically from the time when Jenner got this idea of vaccination, it took almost 100 years to develop a vaccine. Who developed vaccines for chicken cholera and anthrax. We discussed it yesterday how the chicken cholera vaccine came into being, and also how the anthrax vaccine came after experimenting with the sheep. From the late 19th century, vaccines were considered a matter of national prestige, and it became compulsory that there was a compulsory vaccination laws which were passed. In 1885, the French microbiologist Louis Pasteur along with Amélie Roux developed the first vaccine against rabies. Now, in the most importantly later in our 20th century, in 1954, Jonas Salk introduced an injectable vaccine containing an inactivated virus to counter the epidemic of poliomyelitis. Subsequently, Albert Sabin made great strides in the fight against this paralyzing disease by developing an oral vaccine containing a live but weakened virus. The oral vaccination polio vaccine we take during the pulse polio abhiyan is actually a oral vaccine but live vaccine but highly attenuated. Now, why should we vaccinate? What is the need to vaccinate? The immunization has dramatically reduced the incidence of a number of deadly diseases. For example, a worldwide vaccination program resulted in the global eradication of smallpox in 1980, and in most developed countries, immunization has essentially eliminated diphtheria, poliomyelitis and neonatal tetanus. So, coming to the types of vaccines, as I had been just telling you that there could be any types of vaccine. It could be whole organism vaccine where the organism, the pathogen is in a killed state. So, it cannot do any harm, or it has been highly attenuated or weakened due to chemical treatment or rays treatment, UV rays treatment or whatever treatment, but it is highly weakened. So, it would not cause any disease in the person who has been given vaccination. Or it could be purified macromolecules as vaccines, for example, toxoids, capsulopolysaccharides or recombinant microbial antigens or surface antigens. It could be a recombinant vector vaccine, it could be a DNA vaccine, all these are third generation vaccines, or it could be a synthetic peptide vaccines or a multivalent subunit vaccines. We will come one by one to each of the types of vaccines. Now, coming to the whole organism vaccine, as I just told you, it consists of inactivated, killed or live, but attenuated, or we also call them as avirulent bacterial cells or viral particles. The killed or inactivated, some vaccines contain killed, but here the pathogen was previously virulent, it was not avirulent, but it has been killed now. So, it can be killed due to chemical treatment, heat, radioactivity or antibiotics. It cannot produce a disease, it is in a killed state, but it was initially or it was previously virulent. It could cause disease if it had been live or attenuated. Some vaccines containing live or weakened microorganisms, many of these are live viruses that have been cultivated under certain under such conditions which are not ambient to the growth of the organism. So, these vaccines are highly weakened. Now, if you see examples of whole organism vaccines, you will see when we are talking about bacteria being used as a vaccine or a whole organism of bacteria is used as a vaccine. For example, in case of anthrax, it is an inactivated or dead organism which is used as a vaccine. Similarly, cholera, pertussis, plague, they are all inactivated organism. However, the tuberculosis is actually a live attenuated BCG. For example, Bacillus calmette-guerin is an avirulent strain. It is although alive. That is why if given a BCG injection in the first day of the birth of a child, and if you do not see a development of that lump or at the site of injection, you do not again vaccinate because it is a live virus. You are giving a live bacteria. So, you do not repeat the vaccination even if you do not see a boil which is formed at the site of injection. It has to be done only after the repeat vaccination has to be done after proper investigation again. Again, typhoid is a live attenuated bacterial cell. Now, the viral particles, In case of hepatitis A, again it is a killed virus. The influenza and the rotavirus is again, the polio SARC1 is again a killed virus particle used as a vaccine. However, the measles, mumps or rotavirus are live attenuated. They are live viruses, but they are highly weakened viruses. In the normal circumstances, it will not lead to a disease state in the person who is getting a vaccination. Of course, there are exceptions, people with a highly compromised immune system, if given this attenuated virus, there could be a danger, and here and there we do get certain cases where it does cause problems, but more or less it has been highly successful. Now, the purified macromolecules as vaccines, this is the second type, where for example, here again we have three types. The polysaccharide vaccine. Here we know the pathogenic bacteria is having a hydrophilic polysaccharide capsule. Now, this capsule, this bacteria is highly virulent. Now, if we coat this bacteria with antibodies or complement, the process is called opsonization, then our body's immune system immediately recognizes these antibodies or complements and mounts an immune response against this pathogen, and hence kills that pathogen by mounting a good immune response. So, once this polysaccharide vaccine has come into our body has recognized it, has made a memory of, we have developed a memory cells against this vaccine, against this particular bacteria, and now if this bacteria again comes, we will have a much better response to this bacteria. There is another type of vaccine called the toxoid vaccine. For example, in diphtheria and tetanus, these are the bacterial pathogens that produce endotoxins. The endotoxins are further modified using sometimes like chemicals like formaldehyde. These inactivated toxins, which are called toxoids, are extremely effective because it is against these toxins or endotoxins. Endotoxins are very dangerous in case of, for example, in case of a person who has been inflicted with diphtheria or tetanus pathogen. These endotoxins are the ones which causes havoc. Now, if we modify this endotoxin and produce an immune response, a primary immune response, so in case a person is inflicted with the same pathogen again, he has a memory of this pathogen. The recombinant antigen vaccines, here the gene coding for surface antigen of the pathogen can be cloned and used for vaccine development. For example, genes for major surface antigen of hepatitis B virus, they had been cloned. Now, we know it is not the entire antigen which is important, it is the epitope or the antigenic determinant region or the region within the antigen which is important for mounting an immune response. That region could be a surface protein also. Now, if we can mount an antibody response against a particular surface protein, so that is good enough. We do not need to put the entire pathogen in an organism. So, you just clone that particular gene of that particular protein and use it as an antigen to mount an immune response. This is called recombinant antigen vaccines. The third type of the vaccination which is done, as you can see on the slide, is called the recombinant vector vaccine. These are experimental vaccines which are similar to DNA vaccines, but they use an attenuated, that is weakened virus or bacterium to introduce the microbial DNA. The carrier is weakened. You have a pathogen, its DNA is taken, microbial DNA is taken and put into attenuated or a highly weakened bacteria or virus which is used as a carrier, which goes inside the body. This harmless vector virus can then express the protein which are necessary to promote an immune response. Right now, researchers are working to develop recombinant vector vaccines specifically for HIV, rabies and measles. Now, this next type of vaccine which is extremely important these days is called the DNA vaccine. The innovative DNA based influenza vaccines that do not require replication of the whole influenza virus are being developed and tested. These vaccines will only contain portions of the influenza virus genetic material, like only that DNA which is important, which then instructs human cells to make proteins, or to elicit an immune response to that virus. For example, probably the entire genetic or genes of that pathogen is not important. It is only a part or a DNA or a part of the genetic material which is important. So, that DNA is replicated, it is cloned in a plasmid and it is put into into the individual. This DNA based vaccine is passed through a plasmid or a carrier, injected directly into the host or the arm of an individual and can act as a vaccine. We do not require the entire genetic material of the pathogen here. This is called DNA vaccine. The next type is called the synthetic peptide vaccines, which involve producing the influenza virus chemically without their growth in cell culture or in eggs. The portions of the influenza protein that stimulate the antibody production are synthesized and formulated into a vaccine to stimulate an immune response. Here what they do in the lab, they chemically synthesize those peptides that can act as a vaccine. Of course, peptides are never as good a vaccine as an entire individual. So, peptide or as an entire protein. So, peptides are smaller portions. So, to make it more immunogenic, they generally tag it to let us say an adjuvant or some other material to make it much more immunogenic. These are called synthetic peptide vaccines. Now, this another type of very important and a very new concept of vaccines is a multivalent subunit vaccines. Here what we do is, we incorporate multiple antigens from the same microbe into a vaccine through the construction of solid matrix antibody antigen. As you can see, there is a solid matrix on which there are monoclonal antibodies that has been tagged to. Now, to these monoclonal activities, we incorporate multiple antigens from the same microbe which are of important, and then we put this SMAA, that is solid matrix antibody antigen, into an individual that acts as a vaccine and leads to triggering of an immune response. This is called multivalent subunit vaccines. Now, after having understood the entire types of categories of vaccine, the most important thing which comes is the cold chain. The cold chain is a system of storage and transport of vaccines at low temperature from manufacturer to the actual vaccination site. So, most of the efficacy of vaccine is dependent upon this cold chain. If you see, when you go to a doctor or a physician to get a vaccination done, it is generally written that this person is following the cold chain system. It is very important, because if not kept properly, these vaccines lose their efficacy, then they can also become dangerous also. So, the cold chain system is necessary because vaccine failure may occur due to failure to store and transport them under strict temperature control conditions. Now, this is the immunization schedule. As you can see, soon after birth, the child gets a BCG, the OPV first dose and hepatitis B first dose. It has to be repeated after 6 weeks, the hepatitis B second dose, OPV second dose, DPT first dose, and this immunization process goes on till these days you get new vaccines, so you get new schedules also, and so many of, but this is the basic immunization schedule which has to be followed if once a child is born. There are risks associated with vaccines also. The primary risk associated with vaccines is that most of the vaccines that utilize live organism, these live organisms can cause illness also. For example, if the person is immunocompromised or is in a state, physiological state where his immune system is not that good, then even these live organisms which are used in a very low titer as a vaccine can cause actually a disease. So, they can become super antigen and cause stimulation of the immune system. This is one of the very important thing we have to be aware of. So, generally if you see, vaccination is never given if a child is running fever, if he is having some problem, or if he is some way immunocompromised. Yet another risk is that some individuals may have an allergic reaction to the vaccine, especially vaccines produced in embryonated chicken eggs and in transgenic plants. So, because of some difference that has come up in this vaccine that has been developed in a situation which is not native to your situation. For example, if a vaccine has developed in, is been made in chicken, or if, for example, if we use a hyperimmune sera from a horse, it is not actually the antibody produced in our, it may have some differences from the human. So, the immune, if it is given to a human, the human can produce an allergic reaction against that vaccine. So, research and development that is taking place, WHO have a separate unit for research and development of vaccine. Through WHO's initiative for vaccine research, which is called IVR, it facilitates vaccine research and development against pathogens with significant disease and economic burden, with a particular focus on low and middle income countries. Finding a safe, effective and durable HIV vaccine still remains a top priority for our world. The recent research is approaches for designing a preventive HIV vaccine. There has been a vaccine against a dengue virus. Now, NIH scientists identified new HIV inhibiting protein. NIH scientists have found cause of rare immune diseases like genetic mutation leading to cold, allergy, immune deficiency, autoimmunity. NIH found a gene that play a role in type 1 diabetes also. These are the latest research that has happened in the vaccine field. With that, I would like to end my lecture. Thank you very much.
With this note, thank you, ma'am. Thank you so very much for explaining us in detail about the various vaccines. Now here we have some time left. The only question of mine to you is that can we predict or can we think of the vaccines of the diseases, the most uncurable diseases? For example, if you talk about cancers or many kind of such diseases, can we think of vaccines for them in future?
See, technically cancer, if caused because of a virus, see, most of the time cancers is your own cell going hayway, you know, the trigger could be because of anything. So, it is your own cell, your immune system is actually working for it. There is a separate protocol for taking care of the cancerous cells because it is your own cell which has gone wrong. Here when we talk about vaccines, primarily we are talking about a pathogen which is coming from outside. So, when we are talking about a vaccine developed for viruses, yes, we have viruses, we have vaccines for various viruses like rotavirus, we have against polio virus and all. But yes, there is hope so we can develop some vaccines for the cancer viruses also, specifically the cancers which are caused due to viruses. Yes,
surely,
probably.
definitely we can think of such vaccines. As we had a conversation with the doctor in our EDUSET lecture itself. They talked about the vaccines which are specially available for the cervix cancer. They said yes, so
Yes.
for cervix cancer there are vaccines, but for the other cancers still such vaccines are required to be produced. So with the advent of science and technology, we believe that in near future hopefully we would be having more and more vaccines for ample of diseases. Thank you, ma'am.
I would just like to add, for the cervix cancer also, for all the types probably the vaccine is not there because it's called by papilloma, and for all the types of viruses the vaccine is not effective. But yes, primarily that's a good vaccine that can give a good protection. But of course, with vaccination there is never a 100% guarantee.
With this note, thank you, ma'am. Thank you once again for being with us, and we hope that in near future also we would be conducting more lectures and would be knowing more through you. Thank you. Thank you.